Hi everyone, can you see me, hear me? Yes, can you see me, hear me? I guess yes. So, hi everyone, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. Sorry for starting the session late. Actually, both my systems, they were getting updated. That's why there is a delay. So, good evening, welcome to the session, bache, and it is the part 2 of biotechnology principles and processes. And today we are going to continue from the restriction endonucleases, right? And if you people will support me, definitely we are going to end this chapter today. And tomorrow there will be the practice session. Okay, so everyone quickly hit the like button, and you know, if you are new to our channel, what you have to do, you have to subscribe the channel and share the session link with your friends as well. Invite other people to attend this amazing session where we will be discussing the biotechnology in detail and definitely on the basis of NCERT, right? On the basis of NCERT, we are going to cover it. So welcome to the session, everyone. Welcome. So let's start the class. So first of all, as I said, hit the like button, hit the subscribe button. And guys, if you want to join any batch, basically I'm teaching in Avengers 3.0. And if you want to join it, you can use the coupon code Ambika10. And that's the most amazing thing. And that's my profile on Unacademy platform. And here you can join my courses, right? You can join my special classes and they are free of cost, and the detailed content with question practice is provided to you. So just go to right now, this is what you have to do, right? If you have already subscribed my profile, fine, you are getting the notification for the classes. But if not, quickly do it right now. Just go to the Unacademy app, right? Simply on Google also you can type Unacademy login and then search my name, search my profile, follow that, and here you guys can see my courses. Okay, you can be the part of these classes, and here you have the free classes as well. And tomorrow I am going to take this session on Unacademy platform at 7 p.m. That is molecular basis of inheritance, and we will be practicing last 10 years PYQs. Okay, and if you want, okay, if you people want me to again start molecular basis of inheritance in detail on Unacademy platform, so you can tell me here in this session. So your doubts will be taken, the questions will be discussed in detail. So see you all tomorrow at 7 p.m. of course. Clear, bache? So quickly subscribe to my profile, and now let's come to this lecture. Let's start the lecture, and that is your biotechnology principles and processes. So can we start, everyone? Can we start, everyone? Yes. So you revised it from NCERT or not? Whatever we discussed yesterday, you revised it or not? Tell me quickly. Seriously, can I trust you? Can I trust you? So tell me how many questions you practiced. How many questions you have practiced? 10, 20, 30, or nothing? Or nothing? Chale, so let's not waste the time. Let's start the class. So you know that biotechnology principles and processes, as per which in new NCERT, it's chapter number 9. Otherwise, it is chapter number 11. So there is no change in the content, so you don't worry about it. So in the last class, let's have a quick revision of what we have done so far. And yes, see, the free special class. Free special classes are there for you from November 4th to 6th. So be the part of this learning festival, bache, and you know how to join it. And if you want to join Avengers 3.0, this is the coupon code that you all have to use, right? This is the coupon code that you all have to use. So on Unacademy, you will get different opportunities, right, bache? You can get the test series as well. You can get the detailed courses as well. So here you can see the details, right? If you want iconic subscription, the mock test series, this is what you need to scan, and you will get the details of that, right, bache? Right? And from November 1 to 30th, you have Super 30 test series. Okay, and it's again free of cost. So you can enroll for this test series, right? There is nothing that you have to pay, right? The link will be there in the description box, and these are just, you know, some Super 30 test series. So it's a kind of mini test. Okay, after giving that test, you can check right? You can check that, you can check your level, because all the NEET aspirants are going to participate in such type of test series. Done, bache? So that's what you have to do. So now let's start the class and let's start with the revision. What we have covered so far, let's start with that part. So in the last class, we started with the basics of the biotechnology, right, bache? So when we were discussing the biotechnology, so I told you about the old biotechnology also, the traditional biotechnology and the new biotechnology. So when it is the old and the traditional biotechnology, we are only talking about the natural capabilities of the organisms. What are we discussing, bache? We are only talking about the natural capabilities of organisms. For an example, if you talk about the lactic acid bacteria, right? Lactic acid bacteria, it can convert lactose to lactic acid, then it can give us the curd. You know that, right? You know that process, right? So obviously these are the natural capabilities of the organism. So when you deal with such things, it comes under the old biotechnology. So we have many examples. Now when it comes to the new biotechnology, we talk about the recombinant DNA technology. We talk about the genetic engineering. And who's the father of genetic engineering? I hope you all remember Paul Berg. Okay, who's the father of genetic engineering? Who's the father of Nandini genetic engineering? Paul Berg. That's what you have to remember. This part clear? Yes, everyone. This part clear? Okay. Then, bache, I told you about the foundations of the biotechnology. Right? What are the foundations of biotechnology? So can you tell me about the foundations? So biotechnology is actually based on two discoveries. Right? Biotechnology is actually based on two discoveries. So can you tell me about that? Yes. Right now I'm talking about the foundations or the discoveries. Right? So what are that two discoveries? But the session should be interactive. Okay. The session should be interactive. So please participate here in the discussion quickly. So you know that when it comes to the foundations of biotechnology, the discovery of plasmid, which is the extra chromosomal DNA in a bacteria, and the discovery that restriction endonucleases, they can be used in vitro as well. Clear, bache? Clear, bache? So these two are the foundations of the biotechnology. Right? These two are the foundations of the biotechnology. And when it comes to the principles, bache, so here you'll be talking about the genetic engineering and the chemical engineering. That's what we have covered in the last session. Okay? That's what we have covered in the last class. Then, bache, we started the discussion on restriction endonucleases. Right? This is what we started. Wait. Okay, so we started the tools of recombinant, or you can say that rDNA technology. That's what we started, right? We started with the tools of rDNA technology, and the very first thing is enzymes. Okay, so we started with enzymes. Then, bache, it is going to be gel electrophoresis. What is it? Yes, everyone. What is it? It is gel electrophoresis. So the first is enzymes, second is gel electrophoresis, then comes the vectors. Okay, then comes the DNA delivery system, that includes the electroporation, etc. Okay, and then finally we need a competent host. That is required, right? We need a competent host. Clear, bache? So these are the tools of biotechnology. Okay, these are the tools of biotechnology. So we started with the enzymes, and when you talk about the enzymes, the first category that we discussed yesterday was lysing enzymes. Do you remember that? Lysing enzymes. So they are going to break the cell so that we can extract the DNA. Okay, so that we can extract the DNA. Clear, bache? So can you tell me the name of the examples? This is a quick revision part. We have already covered it. So it is chitinase, it is cellulase, and then comes the lysozyme. Right? Then comes the lysozyme. Remember? Seriously? Sure. Then after that, bache, the endonucleases are, sorry, the nucleases are there. The one that are going to digest your, yes, bache, the one that are going to digest your nucleic acid. So here you have exonuclease and you have endonuclease. So it is the endonuclease that is used in the genetic engineering, and especially it is the restriction endonuclease. Remember, It is the restriction endonuclease. So that restriction endonuclease is also known as the molecular Caesar. What is it? It is also known as the molecular Caesar and the chemical scalpel. Done, bache? Molecular Caesar and the chemical scalpel. So don't forget these basic points, bache. So here you guys can see the 3 basic steps which are used for modifying an organism. It includes the identification of DNA with the desirable genes, introduction of identified DNA into the host, and then finally the maintenance of the introduced DNA in the host and transfer that DNA to its progeny. So these are the 3 steps. Clear, bache? These are the 3 steps. Understood? Understood? Done? Sure. So these are the 3 steps. So next is what? Next, yes. So here in the tools of rDNA technology, we have already discussed this part, right, bache? So that's what we have to continue. We need to continue the restriction endonuclease part. Done, bache? So I have already given you the example as well. You know that the first restriction endonuclease discovered was HIND2. Right? It is the first restriction endonuclease that was discovered. And after its discovery, right, after many years, we got to know that this enzyme, it is dependent on a sequence having 6 base pairs. Remember, having 6 base pairs. That's what we have discussed in the last class. Yes or no? Yes or no? We even need to talk about, but right now, I'm telling you about the tools, na? So even I will tell you about the first rDNA formed. Right? Even I will tell you about the first rDNA formed. Clear, bache? Clear, bache? Right? Who formed that first rDNA? That is recombinant DNA. So I told you about the HIND2 and it will recognize 6 base pairs. That's what you have to keep in your mind. And then in the last class, we were talking about the EcoR1. And this EcoR1, it is discovered by, can you tell me the name? Right? This EcoR1, it is discovered by Arbor, Smith and Nathans. It is discovered by Arbor, Smith and Nathans. Clear, bache? Clear, bache? They discovered the EcoR1 and they got a Nobel Prize for that also. Okay, they got the Nobel Prize for that also. Clear, bache? So you know about the nomenclature, right? You all know about the nomenclature, right? So when it is E, this first letter is taken from the, yes, bache, the first letter is taken from the, we have covered this part in the last class. That's why I'm going fast. Yes. So this first letter is taken from the name of the, yes, bache, the name of the genus. Exactly. The first letter is taken from the name of genus. And you know that it will be written in capitals. Clear, bache? So next 2 letters are from the species name. They are written in small letters. This R is telling us about the strain and this 1 is telling us about the order in which we have extracted, in which we have obtained that particular enzyme from that strain of bacteria. So that's what we have covered in the last class. And moreover, in the last class itself, I told you that we have restriction endonuclease 1, we have restriction endonuclease 2, and we even have restriction endonuclease 3. But it is the restriction endonuclease 2 that is used in, right, that is used in genetic engineering. Clear, bache? That is used in genetic engineering because it will inspect, okay, it will inspect the length of DNA and it is going to cut the DNA. Clear, bache? It is going to cut the DNA and it is going to inspect the length of the DNA. That's what we have covered. Now, bache, there comes 2 points. One is the sticky ends and another is the blunt ends. Right? The next thing that we need to know is sticky ends and then blunt ends. Clear, bache? It is sticky ends and then blunt ends. Are you getting it? That's what we need to know now. Now see, when this restriction endonuclease, I told you that restriction endonuclease will make a specific cut. It will recognize a specific sequence, and we used to call it as palindromic sequence. Which sequence it is? It is the palindromic sequence, yes or no? Which sequence it is? It is the palindromic sequence. That's what we have discussed, right? So if the enzyme is eco R1, again, it is also a previous year question. Fine, it is also a previous year question. If the enzyme is eco R1, then this enzyme will recognize this sequence GAATTC. Clear, bache? This enzyme is going to recognize this sequence GAATTC. Are you getting it? Are you getting it? So it's a palindromic sequence. You know the meaning of palindrome, right? Like let's say you have the word madam. So if you are reading it from this side to this side, you will get same spellings. If you will read it from this side to this side, again you will get same spellings. So that is what a palindrome is. Okay, that is what a palindrome is. And now when you're talking about the eco R1 is going to recognize this particular sequence, and that is GAATTC. So in my language, it is known as GATTC. Okay, it is what? It is GATTC. And another important point is, students, that these sequence, how are you going to read such sequences? In 5' to 3' direction, right? In 5' to 3' directions, always remember that. Now, it is also a question. It is also a repeated question. Okay, sometimes in the paper, they can ask you this question in this way, that eco R1, it recognize dash. Yeah, it recognizes which palindromic sequence. So they will give you the sequence, so you should know about this sequence. And one common mistake that students used to make, they do not focus on 5' and 3'. Let's say if it is written like this, if it is written like this, 3' and the 5', if it is written like this, right, then it is incorrect. It is incorrect. So you know how do I remember? I remember it like this, 5G. We have the networks, no? 3G, 4G, 5G. That's how I remember. So always, right, if it is a GATTC, GAATTC, okay, so here it should be 5'. What should be here? It should be 5'. Clear, bache? It should be 5'. So it is 5G. Understood? It is 5G. So when you mark the option, please focus on that points also. So if instead of 5', if here it is 3', then it is going to be incorrect. So that is the palindromic sequence. Now I'm telling you about the sticky ends and the blunt ends that are formed by what? That are formed by your restriction endonucleases. So bache, for sticky ends, let's take the example of eco R1. Actually, we have two categories here. Some restriction enzymes, they form sticky ends. Some restriction enzymes, they form blunt ends. Now, what is the difference? That's what we need to understand. Okay? 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 So, eco R1, it is going to form sticky ends. So, now what is the meaning of sticky end? See, wait. You know now that this restriction endonuclease, it used to cut the sugar phosphate backbone. Do you remember that? It used to cut sugar phosphate backbone. So when you talk about the sticky ends, they are the restriction enzymes cut the strand of the DNA a little away from the center. The restriction enzymes cut the strand of DNA a little away from center, right? It will cut it little away from the center. Now what is the meaning here? See, actually when you are talking about this eco R1, I'm telling you now that this eco R1 will give us the sticky ends. Okay, so this eco R1 is going to make the cut here. You first understand this part. Right? So in between the guanine and adenine, here also, it will consider the same basis. In between the guanine and adenine, it is going to make the cut. So whenever we use the arrows in this way, right? Whenever we use the arrows in this way. Always remember you are talking about the sticky ends here. So basically what is going to happen, bache? The sugar phosphate backbone in between guanine and adenine, right? Where will this restriction enzyme make cut? It is going to break the sugar phosphate backbone in between the guanine and the adenine. Are you getting it? In between the guanine and the adenine. So when you are talking about the sticky ends or the free ends or the cleaving ends, right, here the restriction enzymes cut the strand of DNA a little away from the center of the palindromic site, right, little away from the center of the palindromic site. But always remember, okay, right? So restriction enzymes, they cut the strand of DNA, okay, a little away from the center of the palindromic site, bache. But remember, between the same two bases, right? But between the same two bases on opposite sides. And again, I'm repeating this. It is going to cut the sugar phosphate backbone. It is going to cut what? It is going to cut, Trisha, sugar phosphate backbone. So that's how it is going to make the cut. In between GA, in between GA. Now see, just look at this position, okay? Just look at this position. I'll show you. So if it is making cut, if it is breaking the sugar phosphate backbone in between, don't you think that this AATTC, it will get separated then? This G is still attached to C. Okay, this G is still attached to C, and this is having just a minute. Yes. So don't you think you are going to get something like this? Don't you think that you are going to get something like this? Basically, what is happening? What is happening here? There will be the, right, the sugar phosphate backbone in between the guanine and adenine. It will be hydrolyzed by this restriction enzyme, right, bache? So obviously, in that case, see, this part will stay together, and this part of the DNA, first of all, they will get separated from each other. Try to imagine it. Try to imagine it. I am cutting, I'm making one cut here, I'm making one cut here. So don't you think you will get two complementary overhangs like this? Right? Don't you think that you will get two complementary overhangs like this? Let me explain. G, here it will, see, because you are breaking the bond in between G and A. So obviously this A will, right, this A will get separated. This A will get separated, of course, and here also you are breaking the bond in between. You are breaking the sugar phosphate backbone in between G and A. So obviously here also this part will stay together and this part will stay together. Don't you think so? Don't you think so? Yes or no? Don't you think so? Yes or no? Yes or no? Now just look at these two segments, right? Just look at these two segments, right? Don't you think that they are the complementary overhangs? They are complementary to each other. Like if I will bring this part closer to this part, right? Just try to imagine this. Ashwath, Priyanka, Nandini, Subhashini, Siyam, Nandini, try to imagine this. If I will bring this portion closer to this portion, don't you think this AA, it will form hydrogen bond with TT, TT will form hydrogen bond with AA. They are complementary overhangs now. This is the language of NCERT, yes or no? This is the language of NCERT, yes or no? Yes or no? So basically when you are breaking the bond in between GA and GA here, so obviously this G will remain, this G will get separated, they all will stay together, right? And here also this G will get separated, they all will stay together, and that's what I'm trying to show you here. That's it, that's all. So this is the meaning of complementary overhangs, right? These are the sticky ends, right? And why do we say they are the sticky ends? Because they can clip, right? They can join with another complementary overhang, right? They can join with another complementary overhang, and this is what we need in the genetic engineering. Actually, when I will tell you about the steps of genetic engineering, we will discuss that first of all we have to figure out the desired DNA, right? Then we have to introduce that desired DNA into the host. Firstly, we have to figure it out. Where is that desired DNA? We need to isolate it. Then we need to insert it into the host, right? We will be needing plasmids for that. We will be needing vectors for that, right? So let's say if with the help of this restriction enzyme, if I'm making a cut here, okay, if I'm getting these two complementary overhangs, so imagine if I will use the same enzyme, right, and if I will use the same enzyme for another organism's DNA, I can make the cut there. I can make the cut there. I can pick up this part. I can pick up another part from another DNA and I can join them, isn't it? I can join them. So that's the beauty of these sticky ends. Okay, so this is what we need in genetic engineering. Are you getting it? This is what we need in genetic engineering, so that we can use this, right? We can use this part and we can join it with our desired DNA. And that's how we form the recombinant DNA. As of now, I'm telling you about the tools. So if you will understand the function of this enzyme here, and then in the steps, bache, everything will be crystal clear for you. Okay? Okay? So sticky ends. These are what? These are the sticky ends. So remember, when the complementary overhangs, they will come closer. So no doubt, hydrogen bonds will form in between, right? Hydrogen bonds will form in between. And this G and A, it will be joined with the help of DNA ligase. Okay, so we have another enzyme also, right, Bache Trisha? We have another enzyme also. What is the name of that enzyme? That is the DNA ligase. We have even studied that in molecular basis of inheritance. So it is the molecular glue. Okay, it is the molecular glue. So ligate means to join. So this ligase, it will form the it will join that sugar phosphate backbone. Okay, it is going to join that sugar phosphate backbone. So sugar phosphate backbone will be hydrolyzed by your restriction enzyme, and the ligase is going to join it all together. That's what you need to keep in your mind, and that is the meaning of the sticky ends. Clear, bache? Clear, bache? So in the paper, they can even ask you the examples. Okay, examples of the restriction enzymes that form the sticky ends. Say one is EcoR1. Other than EcoR1, you have the example of Hind3, right? You have the example of what? You have the example of Hind3. And other than that, you have BAMH1 and Cell1. These are the examples of the restriction enzymes that will give you the sticky ends. Clear, bache? That are going to give you the sticky ends. So another point is, bache, blunt ends. Another point is what? Blunt ends. So, bache, when I was explaining you the sticky ends, I told you that restriction enzymes, they cut the strand of DNA a little away from the center of that palindromic sequence. That restriction enzyme, it is going to make the cut in the DNA a little away from the center, right? But it will consider two opposite bases, two same opposite bases. That's what I said. But when you are talking about the blunt ends, so which restriction enzymes are going to make blunt ends? Right? Which restriction enzymes are going to make blunt ends? So here, bache, what's the point? Yes, so enzymes, they cleave both strand of DNA at exactly the same nucleotide position, right? Some enzymes, they cut both strands of DNA at same nucleotide, right? They are going to cut both the strands of DNA at the same, at exactly the same nucleotide. Wait, I will explain. Right, at the same nucleotide position, right, bache? Or you can say that typically it is the center. Typically, it is the center of that palindromic sequence. And this palindromic sequence, it is also known as the restriction site, and it is also known as the recognition site. right? What is it? It is also known as the restriction site. It is also known as the restrictions recognition site. So I will give you one example. So Hind2 that we have discussed, right? Hind2 creates blunt ends. Hind2 creates blunt ends, but your Hind3, it is going to give you the sticky ends. So that's what you need to remember. Okay, that's what you need to remember. Right, Nazarat bache, there is no need to get confused. I have already posted many strategy videos that you can check, and soon I will post the another one. Okay, so till then, if you are new, if you are not able to understand this particular part, just check the first part of biotechnology and then move for this part. Fine, then move for this part. This part clear? Right, right. So Hind2 is the example. You have Eco R5 is the example. Right, Eco R5 is the example. You have SMA1 is the example. So these are the enzymes which are going to give us the blunt ends. Clear bache? Which are going to give us the blunt ends. Just I'll give you one example. So GGCG. So the sequence here is GGCZ. Okay. So that is for your blunt ends. So when the restriction enzyme is giving us the blunt end, that's how it is going to make the cut. For sticky ends, it was like that. Opposite bases were considered, but here the center of the palindromic sequence, and that is how exactly the same sequence they are going to cut. Okay, so what will you get here? This part. So do you think that you will be able to use it for the genetic engineering? Right? Do you think that you will be that you can use it for the genetic engineering? That type of sequences you are going to get, right? That type of sequences you are going to get. Yes, so it is basically the example of HAE3, that is your Haemophilus aegyptus. Okay, that's the simple example. Clear, bache? So that is also the another example of the enzyme which is giving us the blunt ends. So in the case of blunt ends, this is what you are getting, the 2 strands of DNA. So obviously, right, how are you going to ligate with the another desired DNA? It is going to be a tedious task. Don't you think so? Don't you think so? So in such cases, when when these enzymes are used, which forms the blunt ends, then to these blunt ends, you know, sometimes in template independent manner, another bases are added. Okay, right? So sticky ends are formed by adding additional bases. Fine, bache. So mainly what do we need? We need the sticky ends. Mainly what do we need? We need the sticky ends. So here I have mentioned few examples. No need to remember all of them. Yeah, right. But those, the examples which are given in NCERT, you should not miss this. So Eco R1, Hind3 is also important. BAM-H1 is important, right? So this is the TAC one. So I'll share the PDF and you will get the details. Okay, you will get the details. So see, Haemophilus aegyptus. Okay, so the sequence is GGCC. So here what are you getting? You are getting blunt ends. Here what are you getting? Sticky ends. Here also sticky ends. Your bacillus, right? So that's what you can do. Fine, bache? Fine, fine, fine. Got it? Done? Sure? Sure? So this is all about the sticky ends and the blunt ends. Okay, this is all about the sticky ends and the blunt ends. So do remember the examples and see this from NCERT as well. The action of restriction enzyme. So same enzyme, same restriction enzyme is used to cut the DNA of the vector as well, that is your gene taxi, and same restriction enzyme is used to make the cut in the desired DNA containing host. Clear, bache? So see, the enzyme cuts both DNA strand at the same time. So this is the vector's DNA, right? Right, the vector is something which is, vector is also known as gene taxi. So it is something which will help us to transfer the desired gene into the host. And here what do you have? You have the foreign DNA. Here what do you have? You have the foreign DNA. So Eco R1 cuts the DNA between G and A only when the sequence is present in the DNA. Clear, bache? Only when the sequence is present in the DNA. So see, so it will make the cut here, it will make the cut here. right, in the foreign DNA also. So see, this part of the foreign DNA, we are going to join it with the DNA of the vector. So how that bases will join? You know that there will be the hydrogen bond. Okay, how will it join? Obviously there will be the formation of the hydrogen bond because they are complementary to each other, and finally DNA ligase will form the sugar phosphate backbone. Okay, the DNA ligase is going to form the sugar phosphate backbone here. Understood? Sure? Sure? Okay, so another enzyme that we use here is your DNA ligase. Okay, that is the molecular glue. Okay, this is another category of the enzymes. DNA ligase, it is the molecular glue. Now, bache, other than DNA ligase, we are also having some synthesizing enzymes also, which are used in the genetic engineering. Do you know that? We have the synthesizing enzymes also, which are used in the genetic engineering. Just say, let's say, obviously, the word is synthesizing enzyme. So, they are used to synthesize DNA. They are used to synthesize DNA template, right? They are used to synthesize DNA. So, we have the two categories here, right? The one is reverse transcriptase. The one is reverse transcriptase, and another one is, yes, DNA polymerase. Okay, so sometimes if in the genetic engineering there is requirement to synthesize the new strand of DNA, so these two enzymes are used. One is the reverse transcriptase, right, bache? Another is the DNA polymerase. You know that when it is the DNA polymerase, it needs a template, right? It needs a Ashwath DNA template, right, bache? Govinda, Nandini, Subhashini, Trisha, Aarti. So in the case of this DNA polymerase, it synthesizes the DNA, the new strand of DNA, complementary to, okay, the new strand of DNA complementary to a DNA template. A template will be provided, right? A single stranded DNA template will be provided. So complementary to it, it will add the new bases. But when it is the reverse transcriptase, you know that mRNA will be there. What will be there? mRNA will be there, right? So this mRNA is used to form complementary DNA. Clear, bache? So reverse transcriptase is the name of the enzyme. So it is basically the reverse transcription, right? So in the transcription, it is the DNA that forms the mRNA, but here the mRNA is going to form the DNA, and that DNA is known as your complementary DNA. That's how you write it, cDNA. Clear? That's how you write it, cDNA, complementary DNA. Even that, these enzymes are also used there. And finally, the joining enzymes, which is your DNA ligase. Clear, bache? Finally, the joining enzyme, that is your DNA ligase. So next is the vectors that we need to discuss, and then I will tell you about the gel electrophoresis, right, bache? So next part that we need to cover is the vectors, another important topic of this chapter, right? So I think in the year 2021, I don't remember exactly whether it is 2020 or 2021. According to me, 2021, 11 questions were there from the biotechnology. Imagine, 11 questions were there from the biotechnology unit. 11 questions means 44 marks, right? And even if you can, you guys can check the previous year questions from this chapter. They all are going to be same, right? There is no change in that questions. The same type of questions will be repeated again and again. So here for this chapter, this is what I want to advise you people, that first of all, clear your concept, master your NCERT, solve the previous year questions, then you can answer all the questions that will come in your NEET 2024 examination. Fine. So 11 questions, 44 marks. So don't underestimate these two chapters, and trust me, they are the easiest one. So imagine 40 to 50 marks from biotechnology, 50 to 60 marks from the genetics, right? So totally it is giving you near about 90 marks, right? Near about 90 marks. So I don't think that you should miss these units. Okay, and be a little serious, bache. Fine, be a little serious. Okay, so next is what? Next is the vectors that we need to study. Just a minute. Okay, the next topic is what? Next topic is the vectors that we need to study. So vectors, they are behaving like what? They are behaving like the gene taxi, or you also call them vehicle DNA. Okay, vehicle DNA. So now we know what do we need. See, we need to figure out the desired DNA or desired gene. We need to cut that desired gene. Then we have to pass that desired gene into the host, and we have to provide the conditions, right, so that this desired gene can replicate, it can form its multiple copies. That's what we need, isn't it? Isn't it? That's what we need, isn't it? So now when we have to transfer this gene, right, this desired gene or this desired DNA into a host, when we need to transfer this desired gene or desired host DNA into a host, obviously we need a kind of taxi. We need something which can help us to transfer that. So that something is the gene taxi, and that something is actually known as the vector. Right, that something is known as what? That something is known as the vector, because it can transfer our desired gene into the host, right, into the bacteria, where it can form the multiple copies. That's what you need to remember. And even when I started the chapter, when we revised it, I told you very clearly that these two discoveries, they are the foundations of biotechnology, and one of that is plasmid, right? One of that is plasmid, right? One of that is what, everyone? Siyam, Ashwath, Victor, Subhashini, that is the plasmid. So that's what we need to know about the vectors. So these are the these vectors are the vehicle or the vector DNA. Clear, bache? Clear, bache? So here we have different examples. We'll be talking about the plasmids, the cosmids, the bacteriophages are there. Right, bacteriophages are there. And even if you remember, right, in the human genome project, we discussed the BAC and YAC. Now I told you then that we will discuss it in biotechnology. BAC, YAC. What is this BAC? This is bacterial artificial chromosome. It is again a vector. It's bacterial artificial chromosome. It's bacterial artificial chromosome. Clear, bache? Then comes the yeast artificial chromosome. Right, bache? It is the bacterial artificial chromosome. It is the yeast artificial chromosome. Clear, bache? So these are the examples of the vector, and that's what we need to discuss. And sometimes, you know, the viruses, right, even viruses are used. Do you know that? Sometimes even viruses are used to pass the desired gene from one organism to the another. So that's what we need to discuss. We need to talk about the vectors, and the very first thing is the plasmid. Now let me start. And plasmid and bacteriophage, they are very common type of vectors, bache. Let me start with the plasmid first. So what is a plasmid? Plasmid, you know that it is an extra chromosomal DNA. What is it? It is an extra chromosomal DNA. That's what a plasmid is. Yes, bache, that's what a plasmid is. It is the extra chromosomal DNA. It is the extra chromosomal circular DNA. And when you talk about the plasmid, what's the speciality about the plasmid? You know that in a bacterial cell, we can see, first of all, we can see multiple copies of the plasmid. That can vary from bacteria to bacteria and plasmid to plasmid, first thing. And these plasmids, they replicate, right? They replicate independently from this chromosomal DNA. Right? They replicate independently from this chromosomal DNA. That's what we have studied. Yes, bache. That's what we have studied. Yes. So that's why this plasmid is considered as a vector. So how can we say that something is a vector? This is a term that I have used now, vector DNA, or vector, also known as vehicle DNA, also known as vector DNA. It is a gene taxi. So on what basis I'm saying that, you know, certain DNAs, they are behaving like like something which can transport the desired gene? How can we say that? On what basis? So obviously there should be certain features. Don't you think so? There should be certain features that can help there. Yes or no? This is the topic that we are discussing now. Okay, this is the topic that we are discussing now, and trust me, we will cover each and everything from the NCERT. Okay, the sequence can vary a bit, but we will discuss each and everything from the NCERT. So that's what we are discussing, the vector, which is also known as the vehicle DNA. Clear, bache? So obviously there are certain properties, right, which are making that particular DNA a vector, okay, that are making a particular DNA a vehicle. Okay, so these are that properties. So let's start from this part, the cloning vectors. This is our next topic. Okay, this is our next topic, and what is it? The cloning vectors. So let's cover it from NCERT here. So they are saying, see, plasmids and bacteriophages, they have the ability to replicate within the bacterial cell independently, independent of the control of chromosomal DNA. That's what we have. Independent of the control of chromosomal DNA. That's what we have. Yes, that's what we have. Yes or no? That plasmid and bacteriophages, they have the ability to replicate within the bacterial cells independent of the control of the chromosomal DNA. So mark my word that this is going to be the most important point. This is going to be the most important feature. Okay, now come to this part, bacteriophages. Because they of their high number per cell, they have very high copy number. So bache, before understanding the features of the cloning vectors, you should know about certain terms, and that first term is the copy number, right? The first term is what? The first term is the copy number. So can you just tell me about the copy number? Yes, see, the first term is what? It is the copy number. Now whenever you talk about any of the vector, right, whenever you talk about any of the vector, you can consider any vector. Obviously, why are we selecting that vector? Why are we selecting that particular DNA as a vector? Because it can replicate independently. It is the most important feature. Okay, it is the most important feature of a vector, that it can replicate independently. So when you use the word copy number, we have different type of vectors. Definitely they have different properties. Like certain vectors can contain a DNA of, you know, certain base pairs only, or certain kilo base pairs only. Certain vectors, they can replicate the DNA, right, which is having many base pairs. Okay, so these different vectors, they can copy a certain size of the desired DNA. This is the first thing. Second thing is that these vectors, they can make different, they can replicate. Obviously they are going to replicate, and they can form the copies, right? They can replicate and they can form their multiple copies, but that copy number varies. Just let's say this plasmid in a bacterial cell, right? There is a plasmid in the bacterial cell, and in this bacterial cell it is forming only 4 copies. Means it can replicate only for 4 times. Okay, so the copy number of this plasmid will be 4, right? Let's take the example of bacteriophages. In the bacterial cell, let's say bacteriophages can form 10 copies. So I will say that particular bacteriophage copy number is 10. So copy number is telling us about the number of copies, right? Number of copies that a particular vector can form in a bacterial cell or in a host cell. Are you getting it? That is the copy number, and this copy number is linked with the origin of replication site. That's what I am going to tell you. So as per our requirement, that which DNA, which gene we want to copy, depends upon its size as well, depends upon the number of copies that we want. Accordingly, we select the vector. This part clear? Accordingly, we select the vector. Are you getting it? Yes, Trisha, are you getting it? Right? It depends upon our requirement. For example, I just gave you the example of BAC and YAC. I told you about the bacterial artificial chromosome and the yeast artificial chromosome, and I told you clearly that these 2 vectors, they are used in HGP, that is in Human Genome Project. It is also another important MCQ. Fine, bache, it is also another important MCQ. Clear, bache? BAC and YAC, bacterial artificial chromosome and the yeast artificial chromosome. So why in Human Genome Project these, right? Why in Human Genome Project these vectors were used? Because bache, they can form the copies of a large sized DNA or a large sized gene. They can copy that. Okay, right? If that DNA is in MBs, na, it can copy that. That's why. right bache? That's why. Are you getting it? So it depends upon our requirement, that what do we need. On the basis of that, we decide which vector are we going to select. So see, what are they saying? That bacteriophages are having high number per cell, means they can form their multiple copies. That's why they have high copy number of their genome within the bacterial cell. Some plasmids may have only 1 or 2 copies per cell, whereas other can have 15 to 100 copies. So this thing is basically their copy number. So if we are able to link an alien piece of DNA, what is the meaning of alien piece of DNA? The foreign DNA, the desired DNA that we want to copy, right? The desired DNA that we want to copy. So if we are able to link that alien piece of DNA with the bacteriophage or plasmid DNA, obviously we can also get the copies of our desired DNA. Remember, initially in the NCERT it is written, na, when we discuss the rDNA technology, that there will be the gene cloning, and then there will be the gene transfer. It is given there, yes or no? Yes or no? It is given there. It is given now that there will be the cloning of the gene and then there will be the transfer of the gene. Yes, there will be the cloning of the gene and then there will be the transfer of the gene. So how that cloning will be done? With the help of these vectors, because they are multiplying, they are replicating, they are making their multiple copies. So obviously if I will link my alien piece of DNA or my desired DNA with that particular plasmid, it can be bacteriophage, it can be plasmid, right? If I'm linking it, so obviously when this bacteriophage or plasmid, when they will replicate, even my gene will replicate, I will get its clones, I will get its multiple copies. That's how the copy number helps. And then finally I will transfer that desired gene. Are you getting my point? So that's what they are explaining here. Clear, Ashwath? That's what they are explaining here, that if we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number equal to the copy number of the plasmid or bacteriophage. So vectors used at present are engineered in such a way that they can help easy linking of foreign DNA and selection of recombinants and non-recombinants. This part I'll explain later. Okay, so any doubt up to this part? Yes. So these are the features, and the most important feature, as I said, it is the origin of replication, that is your ORI site. Clear? So students, when I was teaching you the DNA replication, when I was teaching you the DNA replication, at that time I mentioned it now, that it is not like that replication can start from any point. No, it is going to start from a particular point, right? And we will call it as ORI site, right? That is origin of replication. Do you remember that? That is origin of replication. So it is the site from where the replication will start, right, bache? So if there is only one origin of replicant, so it will be monoreplicant. If there are many origin of replicant in the DNA, that will be polyreplicant. So your prokaryotes, right, your bacterias, because they have circular DNA, so in their case only one origin of replication is there, they are monoreplicant. But in the case of your eukaryotic cell, bache, in the case of your eukaryotic cell, there will be many origin of replication. There will be many origin of replication. Clear, bache? So they are going to be polyreplicant. Understood? Okay, so origin of replication is a very important property, right? So a vector should have its own, right? Vector should have its own origin of replication site, because it is going to decide that it is going to replicate. Clear, bache? So this is a sequence from where the replication starts, and any piece of DNA which is linked to that particular sequence can be made to replicate within the host cell. Like, for an example, let's say you have this DNA, Okay, so at this point you have the origin of replication. Now with the help of biotechnology, I have inserted my gene of interest near to this, right? I have inserted this alien DNA, or you can also call it as desired DNA, or you can also call it as gene of interest. So I have just inserted it, okay, near to this origin of replication. So obviously when this plasmid, right, when this plasmid, when it will replicate now, so with it my alien DNA will also replicate. Okay, so with it my alien DNA, Trisha, Soumyadeep, Nandini, it will also replicate. So this is a sequence from where the replication starts, and any piece of DNA when linked to the sequence, it can be made to replicate within the host cell. Understood? Sure? Clear? So this sequence is also responsible for controlling the copy number of the linked DNA, of course. So this is another important MCQ, that origin of replication sites, they can control the copy number as well. Fine, they can control the copy number as well. Fine, bache? Fine. So if you want multiple copies of a desired gene, let's say if I want 10 copies of a desired gene, right, of my alien DNA, so accordingly I will select a vector which can give me 10 copies. If I want 100 copies, so accordingly I will select the vector, right? So it is also important for the vector selection as well. Fine. Next is the selectable marker, right? Next is the selectable marker. It is interesting also, and sometimes it is confusing. What is the word selectable marker? Right, selectable marker. See, initially also I have used this in the NCERT. It is given a transformants or non-transformants. These two terms were given there, no? Transformants and non-transformants. These two terms were given there, no? Ashwath, I don't know what are you talking about. A copy number means the number of copies that plasmid can form, right? Let's say you have a plasmid, right? That plasmid can replicate for 5 times. So its copy number will be 5, means it can form 5 copies, right? It can form its 5 copies. That is the copy number. Now let's say I have a gene, right? And I need the 10 copies of that gene. So obviously I will be selecting a vector that multiplies for 10 times so that I can also get 10 copies of my gene, isn't it? Just let's say you have a piece of DNA, or you have a plasmid. So in that plasmid, you have inserted your gene of interest. You have inserted your desired DNA. And now this plasmid will divide, and that plasmid will divide independently. Okay, that plasmid is going to divide. How that plasmid will divide? That plasmid will divide independently. Means it doesn't depend upon the upon that chromosomal DNA. So when it will divide, right? If its copy number is 5, means it will make its 5 copies. Right, it will divide for 5 times. It will make its 5 copies. Are you getting it? Right? Or you can, so basically it is going to make 5 copies. So it will divide in such a way that it will form 5 copies. So if it is going to form 5 copies, because my gene of interest is also inserted in that plasmid, I will also get 5 copies of my gene. That's all. That's it. That's all. Done. That's it. That's all. So now we have the term that is transformant and the non-transformant. Right, transformant and the non-transformant. Transformant, something which is a transformed. Non-transformant, which is not transformed. Right, literally their meaning is simple. Okay, something which is transformed. And now what is the meaning of being transformed here? See, it's very simple. Let's say you have a plasmid. Plasmid is also a circular DNA. It is the extra chromosomal DNA. It is also the circular DNA. So what is it? It is the plasmid. What is it? It is the plasmid. Now in this plasmid, I am trying to insert my gene of interest. This green is what? This green is my gene of interest. Right? This green is what, bache? This green is my gene of interest. Clear, bache? Clear, bache? Now, the process is not as simple as we think. Right now I am telling you, we will extract DNA, we will separate DNA, we will do this with DNA, we will do that with DNA. Actually, it is not like that, right? One of my friends, she used to say that, you know, how does she define the molecular experiments? Yeah, these type of experiments, as per her, basically we need to mix two liquids altogether. Basically, you have to put water in water, right? Right, while performing these experiments, what are we going to do? We are going to put water in water. That is actually not water, that are the solutions. It's not like that, bache, that you can see that, okay, fine, this is the DNA, let's cut this DNA, let's fix it here. No, it's not like that, right? Let's say you have a solution and you know that in this, let's say you have a liquid, right? You know that in that liquid you have the DNA, you have the desired DNA, or you have the vehicle DNA. So let's say if you have desired DNA, you will put the enzymes, right? Obviously certain change in color or under you know, certain other instruments, you can check all that things, but it is not as simple as we talk that, okay, we will cut the DNA, we will fix it here. It's not like that. It's not something physical ki I will pick it up and I will put it like this. No, of course not. Of course not. I need to perform certain experiments. I cannot see all that things happening. I will just assume when I will get the desired results or the standard results, then I will get to know, okay, this process has been done. Okay, so now what's the point here? The point here is that I want to insert my gene of interest here. So if this plasmid has picked up my gene of interest, if it has picked up my gene of interest, right, how will it pick up? We will also discuss that. You know, different ways. You know about the transduction, you know about the transformation as well, and even after this topic we will also discuss that. Okay, we will also discuss that. So what am I trying to say here? Okay, what am I trying to say here? All I'm saying is that, bache, how will we get to know that this plasmid has picked up our gene of interest? Let's say this gene of interest, this gene is for any antibiotic. Just imagine, okay, this gene is for a particular antibiotic. So if this plasmid, initially the plasmid was like this, okay, so if this plasmid has picked up my gene of interest, so obviously this plasmid will also form that antibiotic. Just imagine, forget everything that you have studied. I'm asking you simple questions, you have to answer me. Okay, all I'm saying is this is a plasmid. Let's say I have inserted my gene of interest here in this plasmid. This gene of interest means this gene is basically for the formation of some antibiotic. So obviously if my plasmid will form antibiotic, although it cannot because it will kill the same bacteria, but just imagine, okay, so if it will form that antibiotic, I can say, right, then I can simply say that, okay, my plasmid, right, this is a transformed plasmid, this is a transformant, because it has picked up the gene, right, it has picked up the gene and accordingly it is giving the result. Yes or no? Can I say so? Yes, can I say so? Yes or no? Tell me everyone, can I say so? That if this plasmid has picked up the foreign DNA, the alien DNA, the desired DNA, so it is a transformant, right? Because now it will give one another protein, right? Now it is going to give one another protein. It is a transformant. What is it, Nandini? It is a transformant, isn't it? Isn't it? And let's say if this plasmid, if it didn't pick the foreign gene, obviously it is non-transformant. So if we have to figure out that which one is transformant, means which one has picked up our gene of interest or which one has not picked up our gene of interest, so we need the selectable markers in our vectors, so that with the help of that selectable markers we can say whether the vector is transformant or non-transformant. Clear, bache? The need for selectable marker clear? The need for selectable marker clear? So it helps us to eliminate the non-transformants and to grow the transformants. Clear, bache? And to grow the transformants. So when you talk about the selectable markers, right, these are the certain genes, like the genes which form proteins for antibiotic resistance. So you can say that antibiotic resistant genes, okay, and it can be the genes that form certain enzymes, जैसे first example that I have given you here is antibiotic resistant genes, right? Antibiotic resistant genes, like let's say tetracycline resistant gene, kanamycin resistant gene, chloramphenicol, etc., right? And sometimes the enzymes forming genes, sometimes what, the enzyme forming genes, like your LAC-Z, it used to form beta galactosidase, na, sorry, it forms beta galactosidase, na, beta galactosidases. So obviously they, right, such genes, right, such proteins which give a kind of unique property to the plasmid, right, which give a property that we can with which we can distinguish, right, such type of genes or proteins are considered as the, such type of genes and enzymes are considered as the selectable markers. Okay, they are considered as the selectable markers. Clear bache? Clear bache? Yes or no? I'll explain you with the help of example also, but tell me, is that clear? Is that clear? Sure? Sure? Are you sure? Okay, very good. Very good. So see this. So they are saying that in addition to origin of replication, vector requires a selectable marker which will help to identify and eliminate non-transformants and selectively permitting the growth of transformant. Fine, bache, that's why selectable markers are useful. So transformation is what? It's a procedure through which a piece of DNA is introduced in a host bacterium, right? Through which a piece of DNA is introduced in the host bacterium. Clear, bache? So normally the genes encoding resistance to antibiotic like ampicillin, chloramphenicol, tetracycline, kanamycin, they are considered useful selectable markers for E. coli. So now I'll add one point here, bache. See, if you will, from this part question can come, right? These statements can make the MCQ. It's very simple. What they have given us, right? They have given us a suitable example. They are talking about the E. coli here, Escherichia coli here. What are they talking? They are talking about the E. coli here. They are saying that if like let's say they are talking about the antibiotic resistant genes, right? Antibiotic resistant genes which can provide resistance against the antibiotic ampicillin, against the antibiotic chloramphenicol, against kanamycin, against tetracycline. And because they are giving us the example of E. coli, so they are telling us that E. coli is not having the genes against these antibiotic. So E. coli, because E. coli do not carry any do not carry resistant sorry, do not carry resistance against any of these antibiotics, so we can use these genes as selectable marker in the case of E. coli. So don't get confused in the paper if they are giving you the option E. coli. So E. coli is not having the antibiotic resistant genes against ampicillin, chloramphenicol, kanamycin or tetracycline. That's what you need to remember. It's important. Okay, it's important. I will tell you how that selectable markers work, but this is what you need to remember. And remember, they have given you the reference of E. coli. Clear? They have given you the reference of what? Reference of E. coli. So this this mechanism, the selection of recombinant, it is given here. Clear, bache? It is given here. So I'll explain it, but do remember that here they have given you the example of the E. coli. Okay, let's discuss it. I've used the word transformation. So you know that this transformation word is also related with Griffith's experiment, or Hershey and Chase. Hershey and Chase experiment is basically the transduction experiment, the blending experiment. But in the case of Griffith, we talk about the transformation. We talk about the transformation, yes or no? Yes, bache, yes or no? Do you know about it? So transformation is what? What is the transformation? Let's say this is a DNA. If this DNA is picking up the DNA from surrounding, how will it pick? That mechanism will also be discussed. So if this DNA has picked up the foreign or the alien DNA from the surrounding, then this DNA is going to be the transformed one. It is going to be the transformed one. So if I will link it with the Griffith's experiment, so what was happening there, right? That R strain of the bacteria, right? That R strain of bacteria has picked up the DNA of the S strain, the heat killed. Remember the last stage where the heat killed S strain is added and the live R strain. But R strain got transformed. How? Because it has picked up the DNA from this S strain. So that is the transformation. So transformation means that a DNA has picked up the DNA from the surrounding, right? It has picked up the DNA from the surrounding, and that's what we are trying with our plasmids. Okay, that's what we are trying with our vectors. So if this plasmid will pick up the DNA from surrounding, okay, so this plasmid will be the transformed one, and yes, it is also the recombinant one. Okay, so that's what we need to discuss now, right? We need to talk about the selectable marker. Now I just told you that when you are talking about a vector, okay, plasmid is what? Plasmid is a type of vector. So no doubt it is having the origin of replication, and it should have a selectable marker also. Selectable marker means any gene which is giving it a particular property, which is giving it a selectable property, right? Which will help us to identify, right? Which will help us to identify whether our plasmid is transformed or not. Clear? Whether it is transformed or not. Yes or no? Yes or no? This part clear? Tell me this part clear. First of all, tell me this part clear, then I will explain you the next point. Okay, so now let's say this plasmid here, it is having a, let's say this plasmid here is having a gene, right? It is having gene. So this gene can provide this plasmid resistance against antibiotic A, right? Resistance against antibiotic A. I'm giving you one example. Please listen to me carefully. Antibiotic A. Just imagine. And this plasmid is also having another gene which can provide it resistance against antibiotic B, right? I'm giving you a few examples. So before proceeding, I'll explain you the cloning sites and then I will come to this example, right? After completing it, you will understand this part in a better way. Okay, the 2 points clear, the selectable marker and the, right, the 2 points clear. Right, the 2 points clear, the selectable marker and the origin of replication. So there is a third feature of the vectors, cloning site, and understand this feature because after that you will understand the role of the selectable marker. So cloning sites are also known as recognition sites, but they are also known as the restriction sites. So in order to link the alien DNA, vector needs to have few or single recognition sites. Now you know that when you talk about the restriction endonuclease, you know that they are very specific. They cannot cut DNA at any point. They need certain specific sequences, right? They need certain specific sequences. Yes or no? Yes or no? This is what they need. So obviously if you are talking about a vector, so that vector should have the restriction, should have the sites that can be recognized by restriction enzymes. Yes or no? That vector should have the sites that can be recognized by restriction enzymes. Yes or no? And that sites are known as the cloning sites. That sites are known as the recognition sites. Clear, bache? That sites are known as the cloning sites. That sites are known as the restriction sites. Clear, bache? Clear, bache? Yes or no? Is that clear? Is that clear? So a vector should have at least one or few restriction sites that can be recognized by the restriction endonuclease so that it can make the cut here and we can insert our desired gene or DNA there. Okay, so that we can insert the desired gene or DNA there. So see, in order to link the alien DNA, vector needs to have few, preferably single. See, it is preferable. Remember, preferably single recognition site for commonly used restriction enzymes, right? It should have, vector needs to have very few, preferably single recognition site. If it will be the single one, na, so obviously that particular enzyme will come and it will make the cut. If there will be more, so obviously if let's say we are using 2 enzymes, or if there are more than 2 sites, so if again it will make the cut, then it is difficult to form that recombinant DNA. Done. So presence of more than one recognition sites within the vector will generate several fragments, which will complicate the gene cloning. So that's why it is written here that preferably single. So in statement based question or in assertion and reason based question, that type of points can come. Okay, right? That type of questions and that type of points can come in your assertion and reason based question. Done, bache? So it is preferred to have a single cloning site in a vector than to have many. Why? Because if there will be single cloning site, we will not get several fragments. We will not get many fragments of that vector. Okay, that's why. This part clear? So ligation means joining. Ligation means the annealing, the joining, right? So the joining of alien DNA is carried out at a restriction site, presence in one of the 2 antibiotic resistant gene. Now they are trying to, what are they trying to explain? So as I said, the restriction enzyme should be there. Now look at this vector. Here we have the example of your PBR322. But that's something very important. Okay, the question will come from this part. They can simply ask you the question from the image. They can ask you anything about PBR322, right? And sometimes, you know, at this points, just say right now here, they have given the enzyme name, no? Instead of that, they can just put A and B. They can ask you what is A, what is B, what is C there. So you should know about it. Clear, bache? So you need to know each and every detail related to this PBR322. Clear, bache? Right, related to this PBR322. Done, bache? So it is a double-stranded, obviously it's a plasmid. It is double-stranded circular DNA. Look at this. So E. coli cloning vector, PBR322, it is showing restriction sites. It is showing restriction sites for HIND3, EcoR1, BAMH1, SAL1, PVU2, PST1, CLER1. Imagine. So the first type of question that can come in your final paper is this, that PBR322 has how many restriction sites, right? Yeah, it is having how many sites, right? How many sites for how many recognition sites? So see, 1, 2, 3, 4, 5, 6 and 7. Isn't it 1, 2, 3, 4, 5, 6 and 7? For the 7 different enzymes. 1, 2, 3, 4, 5, 6 and 7. Understood? Understood? Literally that type of questions can come. So please focus here. Okay, please focus here. And they can even ask you that in PBR322, right, which enzymes are having their restriction sites there. So you need to remember this, right? You need to remember this. And you have one another homework, and I want to see the answer in the comment section, right? Your homework is that you will tell me that out of these enzymes, which enzymes are going to form the sticky ends, which enzymes are going to form the blunt ends. That's your homework. Okay, just say HIND3, it is going to give us the sticky ends. EcoR1, it is going to give us the sticky ends. So that's how you have to tell me about the other enzymes. So that's your homework. Clear, bache? So this is what they have. Then here in this PBR322, you have origin of replication, and then you have ROP, right? Then you have ROP. So ROP codes for the proteins involved in the replication of plasmid. ROP codes the protein involved in the replication of plasmid, and even this is what you need to know. Even it is a previous year question. Even it is a previous year question. Are you getting it? Right? So PBR322 is having the restriction sites for 7 restriction endonuclease enzyme, plus it is having origin of replication, plus it is having this rope, right? So basically this is coding the, it is coding the proteins which are required for the replication of plasmid. Now, when you are talking about the selectable markers here you have 2, ampicillin resistant gene and this tetracycline resistant gene. Ampicillin resistant gene and the tetracycline resistant gene. So now look at this part carefully. See, bache, this is the ampicillin resistant gene. And here in this gene, can you see that there are the restriction sites for PST1 and PVU1? Can you see that? It's not mandatory that in the plasmid everything will be put like everything will be placed like this, that this is the restriction site, this is the selectable marker, this is the restriction site, this is the selectable marker. No, actually the restriction sites or the palindromic sequences, you know that what are they? So they are present in between, right? They are present in between. They can be present in between a gene as well. Like here you can see ampicillin resistant gene, tetracycline resistant gene. So ampicillin resistant gene is having two restriction enzymes, two sites for the restriction enzymes, PST1 and PVU1. And here in tetracycline R, right, here it is also having for this BMH1. Yes or no? Yes or no? Now I'll ask you one question. Let's say I am using this particular enzyme. For an example, let's say I am using this restriction endonuclease. So bache Soumyadeep, this restriction endonuclease will make the cut here, yes or no? It is going to make the cut here, yes or no? And same restriction endonuclease will be used to make the cut in the desired DNA fragment, yes or no? Everyone, please revert here. Quickly, kindly revert here. Please be quick. Quick, everyone. So if I'm using this BAM-H1, so obviously it is going to make the cut here. Its recognition site is present here. So don't you think that it will interrupt the tetracycline resistant gene? Like this portion, right? Let's say this portion is coding for tetracycline. Okay, this particular portion is coding for tetracycline. This gene is coding for tetracycline. Now let's say I'm making a cut in between. Okay, and here I'm inserting my desired gene or the desired DNA fragment. So obviously, don't you think that in that case, I will not get the protein which will provide resistance against tetracycline? If I'm using that BAM-H1, okay, if it will make cut here and here I will insert my gene of interest. So it is obvious, students, it is obvious that this sequence, this gene is interrupted. Now this gene, right, this gene will not form tetracycline resistant protein. Is it clear or not? Or if you have doubt, do let me know. Right, if you have any doubt related to this, do let me know. Is it clear or not? Is it clear or not? Clear? Okay. So let's say, right, so now I need to figure out, right, now you will understand the role of selectable marker. Now I know that I have used this enzyme and this enzyme is having its restriction sites in tetracycline gene. Okay, so it is obvious, right, it is obvious that if my plasmid, if my PBr322, okay, if this plasmid, if it is a transformant, okay, if it is a transformant, then it will not give me the resistance against tetracycline, yes or no? It will not give me the resistance against tetracycline, yes or no, Nandini? Again, I'm repeating this PBr322, I'm using this enzyme. So with the help of this enzyme, I'm making the cut here. Here I am inserting my desired DNA fragment. So if I'm going to insert my desired DNA fragment here, bache, so basically I am interrupting the gene. Which gene? I'm breaking the sequence of gene. Which gene? The gene which can provide the tetracycline resistance to that particular bacteria, isn't it? So bache, this is known as insertional inactivation actually. You know, this phenomena is known as insertional inactivation. You are inserting something which is inactivating something. You are inserting a gene that is inactivating another gene. Don't you think it is the insertional inactivation? You are inserting a gene that is inactivating another gene. Of course it is, right? Of course it is. So if I have used this enzyme, if I have inserted my gene of interest here, so obviously my plasmid is no longer have the resistance against the tetracycline, right? It doesn't have the resistance against the tetracycline. So if I have to check whether it is transformant or non-transformant, I have this I can use this, right? Right? If let's say I will make two cultures, right? What will I do? I will make two cultures. Here in this culture, I have added the antibiotics, right? I have added the antibiotic tetracycline. Okay. Okay, here I have added the tetracycline and the ampicillin. Here I have added the both the antibiotics. Let's say I have added. Just take an example. So if my plasmid is transformant, let's say here you have grown certain plasmids. Okay, so here I'm just giving you one example so that you can understand this phenomena. Let's say here you have 8, right? Let's say here you have 8 plasmids. I'm giving you one example, students. So out of that 8 plasmids, let's say your 4 plasmids are transformant. I'm already giving you this information. This is what you are going to calculate later. I'm giving you this information. Okay, out of 8 plasmids, right, your 4 plasmids are transformant. So if they are transformant, do you think, because here in this culture along with ampicillin tetracycline is present, so do you think that these transformant will survive here? Do you think these transformant will survive here? Transformant means they have picked up this gene. Do you think they will survive here? Yes or no? And I really want to see the responses. Please tell me. Please tell me. Obviously, they are not going to survive here. Why? Because tetracycline is there, and due to insertional inactivation, now they do not have the right resistance against the tetracycline. They are not going to survive here. Now let's say this is a culture where only ampicillin is there, right? This is the culture where only ampicillin is there. So obviously here they will survive. Here they will survive. Yes or no? Yes or no? But again you have to check, right? Again you have to even check the tetracycline part also, right? Right? So basically that's how, fine, that's how you can check, right? That is the role of selectable markers. That's how you can use the selectable markers as well, right? And this phenomena is what? This phenomena is your insertional inactivation. Clear, bache? Insertional inactivation. So I will read it from this part, right? Then you can understand further. See, so for example, you can ligate a foreign DNA at this BMH1 site of tetracycline, right? The recombinant plasmid will lose the tetracycline resistance due to insertion of foreign DNA. But it can be selected out from non-recombinants one by plating the transformants on tetracycline containing medium. Obviously, this is what we did now, Ashwath, right? Right? We grew it. Done? Done? In a medium where tetracycline is added. So transformant, when they are growing on ampicillin containing medium, are then transferred on a medium. First of all, we will grow them in a medium where ampicillin is there. Again, you will transfer them on a medium containing tetracycline. So recombinants will grow in ampicillin. Recombinants will grow in ampicillin, right? Right? They will grow in ampicillin. Genes get inactivated due to insertion of alien DNA. Okay. Done. So that's how it works basically. That's how it works. So basically in tetracycline, where tetracycline is present, they will not grow. But if they have the ampicillin, then they can survive. Why? What happened? Because insertion of one gene has inactivated another gene. That is your insertional inactivation actually. That is also your insertional inactivation. But bache, selection of recombinants due to inactivation of antibiotic, it is a cumbersome procedure. It's a difficult procedure. Again and again you have to make the culture. Then you have to shift that, then you have to shift that recombinant. Then you have to check, isn't it? Isn't it? So we have the alternative selectable marker also. So now you got it, the meaning of selectable marker and how these selectable markers they work, right? Now you got it now, the meaning of selectable markers and how these selectable markers they help, they work. Is it clear? So we have another way also, another alternative selectable marker also, which can also help us to differentiate non-recombinants, means non-transformants from the transformant, and how How? Because they can produce the color in the presence of a chromogenic substrate. So here they are talking about the LAC-Z gene. So this gene gives us beta galactosidase. Okay, this gene gives us beta galactosidase. You know that what is the role of beta galactosidase, isn't it? So this beta galactosidase, it acts on the lactose. You know that lactose is a disaccharide. I hope you can relate it with the operant topic. Right, I hope you guys can relate this part with the operant topic. So beta galactosidase will act on this lactose, will break it into glucose plus galactose. That's what we know. This is what we have discussed when we completed the molecular basis of inheritance. Clear, bache? Now when you talk about this particular enzyme, when this enzyme get its substrate, the chromogenic substrate means color producing substrate. What is the meaning of chromogenic? Chroma word is related to color, right? So chromogenic substrate means which can produce the color. Clear, bache? Chromogenic substrate means which can produce the color. So if this enzyme is present and if it is getting its chromogenic substrate, then you will see the bacterial colonies in blue color, right? What will you see? You will see bacterial colonies having the blue color if this enzyme is present, right? This role of enzyme I just related this. I just related it with the operant part. But in general, I'm saying that this LAC-Z gene, it forms this enzyme, and this enzyme when act on its substrate is a color producing substrate. It's a chromogenic substrate. So bacterial colonies are going to be blue. Now here, this gene is also having a sequence for the restriction enzyme. Okay, even this gene is also having a sequence for the restriction enzyme. So obviously, if I will use that restriction enzyme, that will interrupt this gene sequence. So this enzyme will become inactive. Yes or no? This enzyme will become inactive. Yes or no? So if this enzyme is inactive, the bacteria will not give the blue color. Instead of that, the bacteria will give white color. Right? The bacteria will give Govinda white color. So it is the example of insertional inactivation. Right? It is the example of insertional inactivation, and in comparison to that, antibiotic resistant genes, this method is quite easy. It's not difficult, right? We have no need to grow different cultures. If that enzyme is not there, we will get white colonies in the presence of substrate. If enzyme is there, we will get blue colonies in the presence of substrate. So you can read it here, right? You can read it here. So see, in this recombinant DNA is inserted within coding sequence of enzyme beta galactosidase. This results into the inactivation of the gene and the enzyme will not be synthesized. This is the insertional inactivation. So in general, as I said, the presence of chromogenic substrate will give blue color, right? But if bacteria is transformed, then you will get white color. So white colored colonies are showing that plasmid is transformed. Clear, bache? The plasmid is transformed. Done? So that's what we need to cover. So in the next class, we'll be talking about the vectors for cloning genes in plants and animals. Plus, I will tell you about the examples of the vectors. Okay. So next to it, we will discuss in the next class. Fine, bache. So tomorrow at 8:30 p.m. you have one class, and moreover, tomorrow at 7 p.m. we have one class on Unacademy platform, and here we'll be practicing the previous year question, last 10 years PYQs of your molecular basis of inheritance. Fine, that's what we are going to do. So take care, all the best. I'll wait for your comments, and do revise this chapter from NCERT, and please solve the MCQs as well. So thank you so much for watching.